Hello. This video presentation involves the development of phytochemical scaffold for bone tissue engineering using Osmium sanctum and Loofah cylindrica plant extracts. The main aim of this project is to synthesize and characterize scaffolds incorporated with nanoparticles extracted from Osmium sanctum and Loofah cylindrica plants. The nanoparticles are incorporated in order to increase the microbial resistant properties of the implant. The scaffold is prepared using freeze-dry method, and then characterizing it using SEM-FTIRXRD and antimicrobial studies. This is our plan of action. Step 1, by identifying the correct species of the plant, characterized by phytochemical screening. Step 2, extraction of nanoparticles from the plant by two methods, alcoholic extract and aqueous extract. Step 3, characterization of nanoparticles by SEM, FTIR, UV spectra, and antimicrobial studies. Step 4, synthesis of scaffolds, by freeze-dry method. Step 5, designing of scaffold, using tricalicum phosphate, chitosan, and gelatin. Step 6, characterization of nanosacfolds, using SEM-FTIR antimicrobial studies. Step 7, in vitro studies that includes porosity analysis, degradation studies and cell adhesion studies. The first plant is Osmium sanctum. They are easily available at all places, economic, has a good antimicrobial resistance properties, and nanoparticles are easily obtained. It has a good phytochemical resistance towards Staphylococcus and other gram-bacterias. The next plant is Loofah cylindrica. They are also easily available at all places, economic, has a good antimicrobial resistance properties, and nanoparticles are easily obtained. It has a good phytochemical resistance towards fungal and viral species. Aqueous method of plant extract involves two methods, namely steam extract preparation and crushed extraction method. The basic principle involved in steam extract preparation is high temperature will leads to breakage of plant proteins from the cell membrane. Plant extract and phytochemical are water soluble, so that we can able to get the proper extract when compared to alcoholic extract method. In crushed extraction method, the blunt force breaks the intermolecular stable forces and deforms the structural conformity, leading to relays of plant extract. The first conical flask contains the crushed Osmium sanctum, the second contains the steamed Osmium sanctum, the third one contains the crushed Loofah cylindrica, and the last flask contains the steamed Loofah cylindrica extracts. For standardization, the plant extracts are first mixed with AgNO3, and each sample was subjected to preparation of 3 ratios. The ratios were 9:1, 8:2, 7:3, that is, 9 parts AgNO3 in 1 part plant extract, to form silver nanoparticles. All these extracts were kept under 4 places of incubation. For optimization, which include hot air oven, room temperature, sunlight and ultraviolet radiation for 5, 10, and 15 minutes. This image shows the unclear settlement of nanoparticles in the extract, and the next picture shows the clear settlement of nanoparticles. Now let us discuss about the analysis of the nanoparticles. FTIR is a technique which is used to obtain an infrared spectrum of absorption or emission of a solid, liquid or gas. An FTIR spectrometer have high range of resolution data over a narrow range of wavelength at a time. It's a mathematical process used to convert raw data into actual spectrum. This shows the FLIR spectrum of Loofah cylindrica nanoparticles present in the extract. This table indicates the wave number with chemical components which are present in Loofah cylindrica plant extract prepared by aqueous extraction method. This graph shows the FLIR spectrum of Osmium sanctum nanoparticles. This indicates the wave number with chemical components which present in Osmium sanctum. This slide shows the antimicrobial response of the Loofah cylindrica and the bacteria which used will be gram-positive, and the table shows the amount of the extract and diameter of the region. The antimicrobial response of the Osmium sanctum and the bacteria which used will be gram-positive, and the table will show the amount of the extract, and diameter of the region. The antimicrobial response of the loofah cylindrica and the bacteria which is used will be gram-negative, and the table will show the amount of the extract and diameter of the region. The antimicrobial response of the osmum sanctum and the bacteria which used will be gram-negative, and the table will show the amount of the extract and diameter of the region. Let's discuss about UV spectral analysis of sliver nanoparticles. UV spectra is a light absorption spectroscopy in a visible spectral region. This means it uses light in a visible and adjacent ranges. The visible spectrum measures the transitions from ground state to excited state. The molecules contains electrons that can absorb the energy in form of ultraviolet or visible light to excite these electrons to higher molecular orbitals. The more easily excited electrons, the longer the wavelength of light it can absorb. It measures the intensity of light passing through the sample and compares it to an intensity of light before it passes through the sample. This is the UV visible spectrum of the osmum sanctum nanoparticles. This is the UV visible spectrum of the loofah cylindrica nanoparticles. This is the UV visible spectrum of the osmum sanctum and loofah cylindrica nanoparticles. SEM is scanning electron microscope that produces image of a sample by scanning it with a focused beam of electron. The electrons interact with atoms in the sample, producing various signals that contains information about the sample's surface topography and composition. The electron beam is generally scanned in raster scan pattern. SEM can achieve resolution better than 1 nm. The SEM images of the osmum sanctum and loofah cylindrical nanoparticles are shown. Synthesis of nano scaffolds. Cells are often implanted or seeded into an artificial structure capable of supporting three-dimensional tissue formation. Scaffolds usually serve at least one of the following purposes: allow cell attachment and migration, deliver and retain cells and biochemical factors, enable diffusion of vital cell nutrients and expressed products, exert certain mechanical and biological influences to modify the behavior of the cell phase. The objective is to synthesize a scaffold which could help the osteoblast cells to adhere to the surface and metabolize at a higher rate, decomposes in the body, helps in regeneration of bone tissue at a much faster rate. The scaffold acts as an interim synthetic extracellular matrix (ECM) that cells interact prior to forming new tissue. In nanoscaffolds, cell adhesion, proliferation, and differentiation improved when compared to control scaffolds. Natural healing processes in these dense tissues are limited as a result of the mechanically challenging environment of the damaged tissue and the hypocellularity and avascular nature of the extracellular matrix. Freeze-dry method. Different ratios of tricalcium phosphate and gelatin are mixed in the solvent to yield the scaffold. The steps involved in preparing scaffold are as follows: 99% deionized water.